Hello, BISC1323. This is the beginning of recorded lecture 1-3, starting on the chapter on prokaryotes. So right up at the top, we got to look at this name, prokaryotes. What is a prokaryote? Well, we've been using this figure throughout the past couple of chapters to look at all the three domains of life. And if you look carefully at this, you won't find prokaryote anywhere. Prokaryote is not in any of these domains. It's not any of these domains. So what does this word mean? Prokaryotes is a group that includes bacteria and archaea. Lumped together, we call these prokaryotes. Looking at this, we could see that even though bacteria is a complete branch, archaea is a complete branch, eukarya is a complete branch, this lumping of bacteria and archaea together is not a complete branch. It is not a clade. It is not an ancestor in all of its descendants. It's an ancestor in most of its descendants, but we're having to exclude eukaryotes. Therefore, this term prokaryote is, like reptiles, a paraphyletic classification, a paraphyletic group. And again, we're not supposed to do this. Modern phylogeny and taxonomy is supposed to be based on clades, but prokaryotes is not a clade. So why does it exist? Why is the chapter called prokaryotes? Well, prokaryote is a cell type. Cells that are prokaryotes, bacterial cells and archaeal cells, are cells that do not have a nucleus. So even with modern phylogenetic understanding of these organisms, we could see, oh, they're not in a clade together. You can sort of forgive ancient scientists for looking at these things under a microscope and saying, hey, they look exactly the same. They're simple small cells that don't have a nucleus. Let's lump them together. And that's exactly what had happened. So this is a term that still endures and is still used a lot, but evolutionarily it has no significance. It's a paraphyletic group. Prokaryotes are ubiquitous. That means they're basically everywhere that you could imagine life existing in every ecosystem, and even some places where maybe you couldn't imagine life existing. We'll see in a couple of minutes. So yeah, prokaryotes are basically everywhere. Another quick side journey into some history of the world. We'll do more history of the world in a later chapter, but here I got to put in this point that prokaryotes were the earliest life. The earliest living things on this planet are thought to have been these simple prokaryotes getting energy from hydrothermal vents. Photosynthesis is something that came later. A hydrothermal vent, to read from the key terms, is a fissure in Earth's surface that releases geothermally heated water, and the chemicals from these vents power their metabolism of this earliest life. So this simplest life, earliest life, obviously it got a lot more complex from there, but prokaryotes were the ones that started all this. So I mentioned these things exist in places where maybe you couldn't even imagine something existing. Prokaryotes that do that live in these extreme environments, are called, appropriately, extremophiles. The key term says an extremophile is an organism that grows under extreme or harsh conditions. This can be incredibly hot water, this can be incredibly low pH, incredibly high pH, incredibly cold temperatures, incredibly high salt or sugar concentrations. Don't memorize this table, just know the definition of what an extremophile is, and that it can include these sorts of extreme conditions. Another important thing to note about bacteria specifically is bacteria are very commonly cultured in a medical lab as a diagnostic tool. So if you are in a hospital and you're sick with something, or you have some sort of infection and they're trying to figure out what it is, to try to figure out how to best treat you, they're going to try to grow a sample from your wound or sputum or blood or whatever to try to identify what that microorganism is. So yeah, growing these things artificially in a lab on a dish is a very common thing that is done. However, bacteria are very specific in what they require to grow. They require very specific temperatures and types of nutrients, and we have figured out the perfect growing conditions for the types of bacteria that we like to grow. But there are actually a lot of bacteria out there that don't grow under these normal, common conditions, and we term these unculturable. And we didn't know anything about these bacteria until genome sequencing allowed us to sequence all the DNA in a microliter of ocean water and found, whoa, there's all this stuff in here. We didn't know about it because we couldn't grow it. So we know a lot about the things we can grow, but some of these unculturable bacteria are still kind of a mystery to us. Speaking of being unculturable, almost all archaea are unculturable. And I'm betting that when I first introduced this phylogenetic tree in the first lecture here, we all heard of bacteria. We all know what eukaryotes are from BISC1323, but maybe you'd never heard of archaea before. And one of the reasons why you may have never heard of archaea before is because we can't grow most of them, and therefore very little is known about them. So this chapter is about prokaryotes, which include archaea, but to be honest, most of what we're going to talk about in this chapter is going to be about bacteria, just because we know so much more about them compared to archaea. Moving on, let's talk about the structure of prokaryotes. So we did capsid morphology in the last chapter. We've got 3 common bacterial cell morphologies as well. These are cocci, which is spherical, bacilli, which are these rods, and spirillum, which are these corkscrews or these spirals. So yeah, singular, plural, coccus, cocci, bacillus, bacilli, spirillum, spirilli. I'm not going to trick you with a test question about this. This is just, for your interest, singular, plural. Spheres, rods, and spirals. In addition to shape, which they all have, some of these cells have pili and or flagella. And again, some is important because there are plenty of cells that don't have either of these structures. A flagellum and pili are both defined in the key terms. A flagellum is a long hair-like structure that extends past the plasma membrane and moves the cell. And a pilus, or pili is plural, is a surface appendage of some prokaryotes used for attachment to surfaces, including other prokaryotes. So yeah, they'll use this to stick to things, and they'll use this flagellum to swim around. Both defined in the key terms. Importantly, bacteria are not the only cells that have a cell wall, but their cell wall is unique to them. Bacteria specifically have cell walls made of something called peptidoglycan. Now, within this, there are actually 2 different groups of bacteria based on how they use their peptidoglycan, what their cell walls are like. One of these groups is called monoderms, more commonly known as gram-positive bacteria. The gram-positive is based on a staining procedure, which you may learn more about in the lab associated with this class. I'm not going to get into this too much because it's a lab thing. We talk about a gram stain. But monoderms or gram-positive bacteria have, well, mono means one, derm means skin. They've got one skin. They've got one cell wall. There's their cytoplasm. There's their plasma membrane. Here's a thick cell wall made of peptidoglycan. One skin. And yep, single cell membrane, thick cell wall of peptidoglycan. These monoderms are also known as gram-positive bacteria. In contrast, the other group is called diderms, also known as gram-negative bacteria. Again, it's a staining procedure thing. Di means 2, and derm still means skin. Diderms, therefore, have 2 skins because they have cytoplasm, plasma membrane, number 1, thin peptidoglycan cell wall, plasma membrane number 2. These are the inner membrane and the outer membrane, sort of having 2 membranes, 2 skins. So diderms have 2 cell membranes, inner membrane, thin peptidoglycan, outer membrane, and it's the thin layer of peptidoglycan combined with that outer membrane that give them the structural stability of a cell wall. Neither one of these strategies is better than the other. If that were true, selective pressure and natural selection would kick in. These are both good ways to have a strong cell wall, either lots of peptidoglycan or a little bit of peptidoglycan and an extra membrane outside of that. But you should be familiar with these two major groups of prokaryotes. Don't have sexual reproduction. We'll talk more about sexual reproduction when we get into groups that can do this, but prokaryotes can't. And one of the advantages to sexual reproduction is mixing things up and increasing genetic diversity and having diverse offspring. Diversity is very important for evolutionary fitness. So in the absence of sexual reproduction, prokaryotes instead have three distinct mechanisms for what we can just sort of broadly call genetic exchange, mixing up genetics, getting new genes, that kind of stuff. These three things are called transformation, transduction, and conjugation. Let's go through these one at a time. So we'll start with transformation, the simplest one. And this is showing some piece of DNA in the outside world. It gets into the bacterial cell, and then the cell integrates it into its genome. Now it's got new genes. This is a dangerous thing to do, to pick up some random DNA. You don't know what's on it, and now it's a permanent part of your genome, but it's potentially a high reward thing to do. You could acquire some really useful new gene from the environment like this. So this is a mechanism of one of those three mechanisms of genetic exchange, transformation. Cell takes up DNA from environment. It may incorporate it into genome. It may just break it down for parts and energy, but the genetic exchange part is when it puts it into the genome. The second mechanism is called transduction, and this figure is maybe a little too simple. Here's one from a different textbook. So this looks familiar, right? This is a bacteriophage. This is a virus with this binal or head and tail symmetry, and it's doing its virus thing. It's attaching, it's inserting its genetic material. Now, I didn't talk about this in the virus chapter, but what a lot of viruses will do as part of their infection, especially of bacteria, is they'll destroy the host chromosome, or they'll chop it into small pieces. That's just to try to prevent the host from fighting back against this infection. So that's why the blue bacterial host chromosome is in a bunch of pieces here, as the red viral or phage DNA is being replicated. And here you can see the further steps of the lytic cycle. We've got some replication, we've got some assembly, we've got some egress, and yeah, here's a viral capsid filled with viral DNA ready to go off and infect another cell. But look at this capsid. Look closely at it. It's got blue on the inside. This is a viral capsid that has mistakenly packaged itself with a chunk of host DNA instead of more viral or phage DNA. So it's going to go off. It's going to do its attachment and entry because it's the capsid that makes attachment and entry happen, but there's not going to be any infection afterwards because this is not viral DNA. This is some random scrap of bacterial DNA from the previous host. This cell kind of lucked out in getting the dud phage attaching and attacking it, and now it's got some random new piece of DNA, and it may incorporate it into its genome, thereby acquiring new stuff. So to summarize, transduction is spread of DNA by virus or phage. The virus infects the cell, it then mispackages host DNA into a capsid, it quote-unquote infects another cell, and in doing so, it injects not viral DNA, but prokaryotic DNA into this new cell, and that cell may incorporate it into its genome. Now it's got new genes. Our third mechanism for genetic exchange is called conjugation. And once again, this is maybe a little too simple. Here's a more complicated one. We can see this is based on a plasmid. And we talked about plasmids a little bit in BISC1323, but the genes for doing conjugation are located on this plasmid. So here we've got a cell that has the conjugation plasmid and a cell that doesn't have the conjugation plasmid. So this so-called donor cell, I don't like that word, but I don't get to make up these things. I'll explain why I don't like it in a second, but this donor cell reaches out to the recipient cell, attaches to it via a conjugative pilus. Yeah, we talked about pili earlier. It's a special type of pilus, and it gives a copy of this plasmid running through this hollow pilus into the recipient cell. Now, the reason why I don't like calling this a donor cell, even though that is technically what it's called, usually when you donate something, you lose it, like you don't have it anymore. But what's happening during this donation is there's a special type of replication going on at the same time. So by the time you're done, the donor cell still has the plasmid, but this recipient cell, which is now another donor cell, has the plasmid as well. They both have it. It's replicated as it transfers. And at the bare minimum, this plasmid contains the genes required to make this happen, but this plasmid can contain all sorts of other genes, and this process can transfer all sorts of other capabilities and genes to this recipient cell. So to summarize, conjugation is the direct transfer of plasmid DNA from one cell to another, and that key term's definition of a plasmid is an extrachromosomal, covalently closed circular DNA molecule that may contain one or a few genes. So it's like a bonus optional extra chromosome. This is done through a hollow conjugative pilus, and this plasmid replicates as it is transferred. And importantly, any and all these three mechanisms can allow antibiotic resistance genes to spread. They can spread from individuals within a population, or sometimes if the host range is broad enough, they can transfer from one species to a different species. And certainly through transformation, you can sometimes get very different species acquiring brand new antibiotic resistance genes, way faster than evolving antibiotic resistance from scratch. So great for these bacteria, but very notable and bad for us that they enable this kind of spread. Now, we've been talking about bacteria and archaea. I think we should start to break down how they're actually different from one another, because yeah, they look the same under the microscope, and a lot of bacteria a lot of archaea are unculturable. But what are the real differences between these two? Oh, okay, so there are a lot of differences between these two. I'm gonna simplify this. Don't memorize this table. I just want to call out two in particular that I think are particularly important. One, and I've made a little table here with feature, bacteria, archaea, is cell wall. Bacteria specifically have a cell wall made of peptidoglycan. Whether they're gram-positive or gram-negative, they still have peptidoglycan as their cell wall, whereas archaea, it's something else. And lots of different archaea have different cell wall makeups, but whatever it is, it's not peptidoglycan. That is a bacteria thing. The other important feature I want to call out is RNA polymerase. This RNA polymerase, the enzyme that's responsible for transcription, making the transcription bubble and using the DNA template to build RNA. Everything does transcription. Everything has RNA polymerase. All living things have this enzyme. But as it turns out, bacteria only have one version of RNA polymerase, whereas archaea have multiple different versions of RNA polymerase that they use to transcribe different types of genes. This may sound familiar to you. Back in BISC1323, having multiple RNA polymerases was a feature of eukarya. And if we look closely at this, archaea and bacteria are not a clade, but archaea and eukarya, that's actually a clade. They have a deep ancestor, but this is a complete branch containing the two of these. Again, we're not memorizing this table, but there are actually several things that archaea have, features of archaea, that are closer to eukarya than they are to bacteria, despite the fact that under a microscope they look like bacteria and not like eukarya. But anyway, that's all I wanted to say for this tape. All right, let's talk about metabolism now. Let's talk about energy usage. So bacteria can be, these are a couple of key terms back from BISC1323, but this is some good review here. Bacteria can be autotrophs or heterotrophs, and you may remember this slide. Autotrophs are things like plants that get their carbon from CO2 from inorganic sources. And heterotrophs are things like animals that get their carbon by eating stuff. And yeah, there are some bacteria that are autotrophs, some bacteria that are heterotrophs. They could be either of these, and they're both defined in the key terms. Importantly, these two distinctions are based on where carbon comes from. Additionally, bacteria can be phototrophs or chemotrophs. These two distinctions are based on where energy comes from. Photo, if you couldn't guess it based on the name, energy comes from light. Let me read the key terms here. An organism that obtains energy from sunlight. Got it. Chemotroph, maybe you could guess based on chemo in the name. A chemotroph is an organism that obtains energy from chemical compounds. So these two are based on energy. These two are based on carbon. Predictably, there's a crazy flowchart, and there are 4 possible combinations of these different categories. Bacteria can be photoautotrophs, chemoautotrophs, photoheterotrophs, or chemoheterotrophs. And if your head is spinning from all these terms, trust me, it's just these 4 that we already know that are really straightforward, but put together in different combinations. Photoautotrophs get their energy from sunlight and their carbon from inorganic sources. Plants are classically photoautotrophs, but there are plenty of bacteria that are as well. Chemoheterotrophs get their energy from chemicals, and they get their carbon from fixed carbon, from eating stuff where the carbon is already organic. Animals are classically chemoheterotrophs, but there are some bacteria with this lifestyle as well. Now the two weirdos. Chemoautotrophs get their energy not from sunlight, but from chemicals, and they are fixing their own carbon, but they're not using energy from sunlight to do that. I don't know if you can tell. That's a hydrothermal vent. We already talked about these things earlier. These are chemoautotrophs. And then, in my opinion, the weirdest ones are the photoheterotrophs. This you can find, they're not that exotic. You can find this in mud. Like pond mud. Photoheterotrophs get their energy from sunlight, like a plant, but they're not fixing their own carbon. They're getting their carbon from organic sources. So again, all 4 of these are in the key terms if you want to look them up, but I think just understanding these 4 and knowing how they combine is a faster way to understanding this. Okay, now we're to talk about bacterial diseases in humans. There are a bunch of bacteria that can cause disease in humans. We call these pathogenic bacteria. I'm not going to make you memorize this, but yeah, these are some of the ones that we can call out here. Anthrax, botulism, I'm not going to read them all, but yeah, there are lots of diseases that are caused by bacteria. And again, the important thing here is that we call them pathogenic bacteria. Now, I'm not going to make you memorize all of these pathogenic bacteria, but when it comes to archaea, I think the list is short enough. I do want us to memorize all of the diseases caused by archaea. It's nothing. It's none. It is a blank slide. There are no diseases caused by archaea. No archaea are pathogenic. I mentioned earlier in this lecture that a lot of archaea are unculturable, and that's why we don't know a lot about them. Here's the other reason we don't know a lot about them. There's not a lot of grant funding for studying something that's not actively killing us. We know lots about the microorganisms that are killing us, but we definitely don't know as much about things that are not pathogenic at all. There are no archaea that are known to be pathogenic, not to humans, not to plants, not to anything. There are no known archaeal pathogens. Speaking of disease, though, let's talk a little bit about antibiotics. So, an antibiotic, let me read the key terms definition. An antibiotic is a biological substance that in low concentrations is antagonistic to the growth of bacteria. So it doesn't have to kill bacteria, it just has to make them unhappy. It has to prevent them from growing, and sometimes it does kill them. So antibiotics that we use create selective pressure favoring individuals that resist antibiotics. And I know this was an example I talked about in BISC1323. The reason antibiotic-resistant strains of bacteria exist is because we use antibiotics, and that makes selective pressure, and that's how evolution is driven. I'm not saying don't take antibiotics. If you've got a bacterial infection and you're prescribed an antibiotic, you take the whole thing. But overuse of antibiotics has created strains of bacteria that resist these antibiotics. Sometimes 1, 2, 3, 4 different types or categories of antibiotics. It's becoming a big problem. There are many different ways that bacteria can evolve resistance to these antibiotic drugs. The most straightforward way is that they evolve an enzyme that destroys the antibiotic itself. Or maybe they evolve pumps that get rid of the antibiotics so they're not affected by it as much. Maybe they stop allowing the antibiotic to enter their cell and then they don't care about it anymore. Or maybe the thing that it shuts down, they just find a different way to do that important thing, and so they don't care about it. So there are lots of different ways to evolve antibiotic resistance. How do we get around this, though? Well, one solution is to make novel antibiotics. But won't they just evolve resistance to these new antibiotics? Yes, then we make new antibiotics. And so this is in some ways like a treadmill. Welcome to evolution, welcome to nature. Everyone deals with this treadmill. But yeah, one solution to this antibiotic resistance is to come up with new antibiotics, especially antibiotics that work in novel ways. Another solution to this crisis of antibiotic resistance is to develop vaccines that prevent infection instead of trying to be antagonistic to the growth of the bacteria. Allow your immune system to fight them off and help your immune system do that with a vaccine. Again, I mentioned this in the virus chapter. We'll talk way more about vaccines near the end of the quarter in the immune system chapter, so look forward to that. Now, I don't want to just talk about bad bacteria and horrible diseases and bad things happening to us. There are actually quite a few beneficial prokaryotes. A lot of these beneficial prokaryotes make their home in and on us. So this is referred to as the human microbiome. This is a very short list of some of the bacteria and fungus, that's getting into a different chapter though, that routinely live on our skin. And there's a whole ecosystem of different bacteria that live in our digestive tract as well. Again, part of what's called the human microbiome. These microorganisms don't typically harm us. They're not considered to be pathogens, but some can cause harm if they gain access to other locations of the body or gain toxin genes. So a lot of these things living on our skin will become a problem if they gain access to other parts of the body. And some of these in the digestive tract, E. coli, harmless, but if it gains the Shiga toxin gene, then it's going to be a problem. Then it is going to be pathogenic. Side note, where would it possibly get the Shiga toxin gene? Oh, I don't know, maybe one of these mechanisms of genetic exchange where it picks up that gene from the environment or from a phage. And so there are definitely ways to make these non-pathogenic bacteria in our microbiome suddenly become pathogenic. So this kind of makes it sound like they are a liability if they're not causing harm, but they could cause harm at any moment if something bad happens. Well, that doesn't mean we want to get rid of them, because they actually actively help us in many ways. On the skin, these flora help crowd out pathogens. So they sort of take up a lot of the available real estate on our skin and make it very difficult for pathogenic microorganisms to try to cause a skin infection. It's not a perfect barrier, but it does a pretty good job. And in the gut, these microorganisms play a very important role in helping us break down parts of our food and create vitamins for us. So they may be somewhat of a liability, but it's definitely not something that we want to get rid of. They're definitely very beneficial to us. Phylogeny. So, okay, sorry, I should have gone to this. We've got this phylogenetic tree of bacteria. If we want to expand this, it gets really complicated. In my opinion, too complicated to talk about in an intro course. Bacterial phylogeny is complex, and it's difficult to assign. Remember this horizontal gene transfer we talked about in the first lecture? That happens a lot with bacteria through those three mechanisms of genetic exchange, and makes it very difficult to draw these sorts of trees with any sort of accuracy. They obviously exist, but you can learn more about those in a more detailed microbiology course. All I want to say about phylogeny here is that it's complex and difficult to assign because they're doing horizontal gene transfer all the time. Okay, that brings us to the end of this prokaryotes, although it was mostly bacteria, prokaryotes chapter, and also the end of recorded lecture 1-3.